Hello and welcome to today's webinar on Metabolon's integrated bioinformatics platform. My name is Heino Heyman and I'm the Global Applications Lead at Metabolon. It's an absolute pleasure to have you all with us today. We're here to explore some of the advancements in the field of metabolomics and how these are shaping our understandings and capabilities in life science research. Joining me today is my esteemed colleague, Joey Garner, who is our principal bioinformatician in our bioinformatics team, and who has been integrally involved in developing this bioinformatics platform that we will be presenting today. The expertise of our bioinformatics team has been crucial in pushing the boundaries of what's possible in bioinformatics, contributing significantly to the platform's innovative features and robust performance. We're excited to share with you the insights and functionalities of our new bioinformatics platform, designed to enhance your research and analytical capabilities. So some of you in the audience today who tuned in to hear more about the bioinformatics platform might be thinking, who is Metabolon and why are they developing new bioinformatics tools? So Metabolon is the global leader in metabolomics analyses. With 24 years of leadership within metabolomics, we have been providing leading expertise in biochemistry and its role in health, wellness, nutrition, and diseases. Depending on the need, we provide a suite of panels and assays to measure and quantify biologically relevant metabolites, small molecules, and determine their relevance to each of our clients' biological questions. We have developed unrivaled capabilities on our analytical platform, our metabolite database, our advanced curation process, and our biological interpretation. Metabolon has grown its capacity, its technology, and knowledge as the metabolomics field has advanced and has become more mainstream. At Metabolon, we have been doing metabolomics quite some time, as I already mentioned. Over this period, we have done more than 10,000 projects. We have profiled more than 200 sample types, 100 plus different species, and investigated more than 500 different diseases. And overall, we have analyzed more than 1 million biological samples. The bottom line here is, if you're investigating something, the likelihood of us having investigated it as well is quite high. We have worked with clients from all over the world on projects big and small, multi-centered and large population health studies across a spectrum of species, and have a broad experience and understanding of many different application fields. This really enables us to provide the highest level of quality, coverage, and comprehensive analysis for our clients. You might be asking, why is this important? We intimately understand the needs of researchers because we ourselves are researchers. And as we have grown, we have developed significant capacity and are now able to process more than 300,000 samples globally each year, matching each sample against our industry-leading 5,400 metabolite database, covering more than 70 different biological pathways. Metabolon has contributed to over 3,500 publications. Clients often invite us to collaborate and be co-authors on the research, but even when that is not the case, people are very happy with our data. They explicitly state the aspects of the study that was performed by Metabolon. So Metabolon really provides an end-to-end service, providing personalized guidance at every step, from study design to sample collection, report visualization to biological interpretation. Metabolon experts guide you every step of the way. When we talk about study design, this is of course very important to chart out the best course for a study and making sure that it adheres to everything necessary for a metabolomic study. Sample prep is done internally at Metabolon. This really allows us to build a strong foundation for any research that we are doing. The analysis that we provide is at the highest level, really to ensure precision and reproducibility for any project you do today and any follow-up projects you do in the future. Curation is a cornerstone of our analysis. This is where we use AI and machine learning to take our data and identify metabolites and let them rise above the noise. When we get to bioinformatics, here's where we really harness the power of our historical insights. And when it gets to interpretation, which we are very proud of, this is really where we take results beyond a data table. Our discovering translational scientist experts elevate the meaningful biological insights from the pages of spreadsheets and really allow you to delve deeper into your data. At Metabolon, we follow two specific philosophies. The first one is a chemocentric approach that we have. That means that we annotate our metabolites and we provide those to our clients, that the set of metabolites that we can confidently annotate. What that level 1 identification means is that we matched a metabolite within your data set against the reference standard run on our platform under the exact same conditions. On the right-hand side, this speaks to another philosophy that we want to give biological context. To each metabolite that was identified in your data set, we will assign it to a chemical class or biological pathway. So this allows you to look at your data not only at numbers, but also biological context. Interpretation support has been a staple of Metabolon service offering, helping researchers bridge the gap between data that they have acquired and the insights they garner. And this is something that we are especially proud of. But we know that to accelerate the maturity of metabolomics research, we need to put the power into the client's hands. Some tools have been available for a long time as part of our project deliverables, but they still required users to take their data file and investigate it beyond the individual reports provided as part of our interpretive services. Getting accessibility and power into our researchers and our clients' hands is of paramount importance to us at Metabolon. And taking what you see here as our previous solution and delivering a high utility integrated bioinformatics platform to empower you to dig deeper. This is good, it's valuable, it's been widely used, and it was intentional. But we are not complacent, and as people become more familiar with metabolomics, they want to do more with it. So this is really the jumping off point for a new solution for accessible, empowering bioinformatics tools. We will now walk you through the new bioinformatics platform that we have at Metabolon. A guiding principle for this new bioinformatics tool has been to combine our extensive industry experience with cutting-edge technology. We are empowering our researchers and clients going forward to navigate complex data effortlessly and accelerate their discoveries in their life science research. So at Metabolon, translatable data is key. And for any project to be to really have an important component, it needs to be translatable. The new bioinformatics platform from Metabolon showcases this exactly. To take our clients from data to biological insights as quickly as possible. With large volumes of data becoming the new norm, high quality data generation is only one piece of the puzzle. But to get translatable insights from this data remains an industry-wide challenge. Delays due to inconclusive outcomes are very costly and can delay life-saving therapies to be discovered. With that said, I think researchers everywhere would agree that the right bioinformatics solution to elucidate your findings has never been more important when it comes to designing a study. So it's now our pleasure to introduce Metabolon's integrated bioinformatics platform. This platform is powered by the Metabolon's untargeted metabolomics technology generated by our global discovery panel. And it has been developed with an extensive set of expertise from our bioinformatics team, from our translational team, to really provide this to our clients. Clients now have this platform to interrogate Metabolon's data generated by us as they have never done before. All levels of expertise can use this tool. If this is your first metabolomic study with Metabolon, or if it's your 100th study with Metabolon, all of that range of expertise can all use our new tool. It's one platform that easily moves from one tool to the next. So the last 20 years, and this statement might seem I'm repeating myself, but it's really the message that we want to bring. The last 20 years have seen tremendous advances in metabolomics, and many of those have been supported by Metabolon. And with its ability, with metabolomics' ability to scan large numbers of metabolites and provide a snapshot of an individual's health status, Metabolon has been in the forefront of this growing field, setting new benchmarks in metabolomics excellence for over 2 decades. This pioneering journey has consistently expanded the boundaries of expectation, offering unparalleled depth and breadth in metabolites analysis from a single biological sample. Our technology ensures that the richness of the high quality data that is given and shifting the focus towards unlocking novel insights and deepening the understanding of your data. The need to decipher biological data from high dimensional data has led to the creation of this integrated bioinformatics platform. Designed with precision for scientific exploration, our platform empowers researchers with immediate access to tools for dissecting complex biological questions and fostering an environment to gain deeper insights into the data. So going forward, all data provided from Metabolon's global discovery panel will be available as a complementary service within our integrated bioinformatics platform. It is very important to highlight this as well. The computational power of Metabolon's bioinformatics platform gives clients the confidence they need to work with large data sets. Big data is not a problem in our platform. It has been tested and validated with tens of thousands of samples. The platform is capable of quickly rendering your data in visual tools in real time, as well as giving you the flexibility of working without needing to reconfigure or re-upload data sets to continue your analysis. Multivariate analysis tools require robust validation. This has also been something that that is very important to Metabolon, to be able to validate the bioinformatics results that you are getting, to be confident in your results. But Metabolon's integrated bioinformatics platform enables customers to do this easily without needing to code additional lines of Python or R to get the data you are seeking to understand most. It is possible to achieve the same outcomes in other platforms. However, they are usually slow, unintuitive, and typically require the user to have advanced knowledge of statistical analysis and coding languages like R or Python. Metabolon's integrated platform is fluid, easy, and doesn't interrupt your investigation to prepare new visualization formats. So, without further ado, I'm excited to hand over to Joey to give us an overview of the platform and how the tools work. Over to you, Joey.
Thanks, Heino. Yeah, and appreciate you all coming to see this platform. And just to echo some of Heino's points, is we really want to put a fine point on showcasing the exceptional and best-in-class analytical chemistry approaches that we have. So hopefully this showcases that, as well as gives you a lot of touch points as a user determine the best outcomes for your data as quickly as possible, and make as many decisions and progressive decisions as you do so. Another intention of this is to give you some more access to different ways that you can look at the data, and spend more time researching it, because I know for some of these multidimensional data sets, it can be there can be a lot of richness, but it can take a lot of searching. So I think one of the best ways that we can exemplify the general utility of this platform is with a pretty simple use case. So in this case, looking specifically at PCA, which is, I think, pretty something that most people are familiar with. We've tried to create an integrated atmosphere where you can look at the data a couple of different ways and form a narrative off of that. So we have 5 tabs that I'll walk you through in a second here, including the overview with the traditional PCA scores that you can look at several different dimensions, as well as a PCA plot, a scree plot showing the variance as you progress in dimensions to see where the majority of the variance is being explained, as well as a loadings plot to start to see what specific features and metabolites are contributing to the dimensional decomposition, and then the biplot that is an overlay of a couple of those things. So to kind of walk you actually through this, one thing that I'd like to introduce is we're going to be looking at a basic a simulated inflammatory bowel disease data set. So I'll make some references there and kind of give you an idea of how you might interact with the platform to gain some findings from it. So you might see first off a really traditional plot of the PCA where we've dimensionally reduced the data and might want to apply some different ways of looking at it, including coloring by gender if we want to investigate some particular confounders in our data set. One thing that's really important to us is that we want to give you all of your data at any point. So I just went past the data table there, and you can download that at any point. Customizability is also really important to us. So if there's a color scheme that you prefer, as well as a confidence interval that you'd like to put on, we're trying to make this as accessible to you so you can download the plot and put it right into your figures or presentations of interest. All of our plots are interactive, so you can drag and drop, you can interact and add different principal components, and scale it to 3 dimensions. Like I said, there are a couple of other tabs, including the scree tab, so you can look at elbow points for where the variance might drop off, and other qualitative metrics to see how well your data are explained, as well as the loadings plot for the individual metabolites. We have this powerful biplot, which is kind of an overlay of a few things, so you can start to already within the first 5 minutes of interacting with our platform, start to gain some insight into what your particular samples are doing, as well as what metabolites are doing. And this is really useful not only for diagnosing what's happening in your data, but also quickly understanding if the outcome is consistent with your experimental design, and to figure out if there are any batch effects or any other QC criteria that you need to be aware of. This is, like I said, available for all data sets immediately, so it doesn't matter how big your study is. You can apply this to really anything, which for people like me who have tinkered a lot in PCA can be quite powerful, because even an hour back a day is really useful. So moving on to the next page that I'll be talking about, it's partial least squares discriminant analysis. And this is a fairly popular technique within metabolomics that serves to use insights onto which specific groups each sample is in, and to try to derive the metabolites that are driving the differentiation between those features. So in a study like this, where we have an inflammatory bowel disease cohort of patients, and then we have some healthy patients, this is a good way of describing the specific metabolites that are driving the differences between those 2 sets of patients. And this is really the first step on our investigation into biomarkers, and particularly the metabolites that represent these 2 phenotypes. One thing that I'd like to point out here too is that we've really tried to make this as consistent an environment as possible as you go from page to page. And we really want to lower the barrier to entry for all sorts of users, give them the access to the data in a way that they can make decisions off of. But if they want to do further studies, if they want to take the data and apply more machine learning to it, then it's your data, and we want you to have access to it. So you get like I said, a bird's eye view of the component interactions, and it looks a lot like a PCA plot. But when I show this to you, we'll spend some time looking at the feature importance plot and how that might inform the specific metabolites that drive the interactions between biology and metabolism. So when I press play here, you'll see it looks quite a bit like the PCA. You have a lot of the same customization parameters. You can download any of these plots. You can add symbols if you, again, want to look at confounding variables that might also be driving the groups. We have, again, this customizable plot in 3D if you want to interact and look at things in higher dimensions. But I think where the power starts to begin with PLS-DA is a plot like this, where you have the importance of each of the metabolites in compared to the outcomes. And you can click and interact directly with each of the metabolites and see the actual distributions of the metabolites in each of the groups. You can scale up and look at a whole different set of metabolites without doing any sort of data engineering or interaction on the back end. And this is a no-code environment, so it should be open to everybody. We can again see the loadings as well as the biplot to look at the specific metabolites that are driving these features. If we have an a priori metabolite that we'd like to investigate further, we can plop that right on the biplot immediately and zoom in to interact. And you can see something like methylsuccinate seems to go with these other amino acids. We're also sensitive to the fact that PLS-DA can overfit data quite easily. So we're giving you access to cross-validation and permutation testing statistics so that you can really dive deep into knowing whether the data actually represent a phenotype or are better descriptive of the specific study and start to parse out some biomarkers that way. So far I've talked about two-dimensional reduction techniques, but we also are really sensitive to the fact that it's often very nice to just look at your statistical results in a way like a Volcano plot that you should be really familiar with. And I'd like to pause here and mention that, within Metabolon, we have a history of having some really talented statisticians and some really talented translational folks that are working really hard to get your data into a format that best represents it in a statistical reasoning perspective and is given to you in a way that you can make the most reasonable deductions from. So with the Volcano plot, we're using the statistics that we're building in-house and running based on our experience and sending to you based on the study design. So it's all powered by that. And then we have a Volcano plot, which is a really familiar way to view that. This is paired with a heat map that is available, and it's consistent for our existing users with the heat map that was provided in the client data tables historically. And as I progress forward, I think it should be quite familiar how this Volcano plot works. So again, you have the significance bars on the right and the left, and you have the p-value, the fold change on the right and the left, and the p-value on the bottom. And you can directly customize that in the app as you'd like, if you want to make it less restrictive or more restrictive. Again, if you want to look at the individual values of any of these metabolites, you can scroll over and interact with them that way. If you click on them, the box plot will appear. Or again, if you have a specific metabolite of interest that you'd like to investigate, you can star it immediately. You can add all these other customization parameters, anything that makes sense for you to build this out so that you can directly put it into a slide deck and make the best use of your data. That's kind of our guiding principle here. I think something that's starting to hint at where we excel is we have all of these metabolites contextualized. We're aware that these metabolites can be confusingly named. They can kind of fall in a lot of different biological principles. So we can do things like search by pathway and send that directly back to the Volcano plot. And you can see here that all arginine metabolism metabolites are highlighted here. So you can directly see what a single pathway or what a single hypothesis might be providing for you as you interact with a Volcano plot. And I'll revisit this in a little while, but I think something that really sets us apart is the ability to re-enter the app, cut the data different ways with the solid foundation of great analytical chemistry, great curation, and great statistics. The last thing that I'll show you before handing back to Heino, and kind of within the first couple of tools that we think are really representative of the vast majority of statistical features that are important for people directly interacting with their data on a day-to-day basis, is this idea of hierarchical clustering. So clustering analysis can be used for a lot of different reasons, and unsupervised clustering especially is quite powerful for picking out things from your data that might not be expected or might not be predicted. We can also use it, as simply as for quality control or assessment of study design. So there's a lot of powerful utility behind using clustering analysis. So this is kind of the mock-up of the page, and it will look really very similar to this, with a lot of the customization parameters that hopefully you've come to expect by looking at the previous 3 pages. It also comes with 3 separate tabs, which I think are all quite powerful and advanced in their own right, and all, again, are no-code solutions and quite fast and performant. So we have clustering analysis, where we have the typical hierarchical clustering, as well as embedding using some dimensional reduction techniques that are great for diagnosing clusters, as well as correlation if you want to start to see what some of those clusterings are doing in contrast or in concert with each other. So now that we move on to this page, it looks fairly similar now. We have this hopefully cohesive looking framework. And as I press play, you can see I scroll down, I see this big heat map, I see some patterns starting to surface, but I might be interested in first seeing if my experimental design worked out. And I see that my samples really do cluster very closely together. So the IBD patients are very different than the control patients. If I go back to metabolites, I can change the way that the cluster is forming by adjusting the p-values or the fold change thresholds. I can lower or heighten this dendrogram bar to make the clusters more or less granular. And then I think a powerful feature is that this is completely interactive. So you can see the identities of the particular clusters as you interact with it. We can send this back to the Volcano plot, because I think the Volcano plot is a great home base for discovery. And you can start to see that there are elements of that that seem differential, but then there are some things that just don't seem that differential. We can look at the specific cluster in detail and interact with it there, as well as send it to this clustering embedding plot. So if we want to look at something in the popular UMAP technique, we can do it there. And we can start to see how different metabolites are forming these patterns. If we scroll down to the bottom, we can again see different clustering techniques and different unsupervised clustering techniques and how they form different clusters. And the last page is, we feel like we're starting to discover some patterns of the data, some subphenotypes that are not quite full phenotypes, but they're not individual metabolites. And we want to see how those groups of metabolites are performing in contrast to other metabolites. And we can do that with this cluster correlation. We have, again, a lot of these aesthetic parameters that we can adjust, and then we can mouse over any of these things to see what the contents of the clusters are. So this is, I think, a really powerful tool for unstructured exploration into your data, and I think there are a ton of ways to think about it from here. So I think, with that, I'd like to hand back to Heino so that he can mention some of the finer points here.
Thank you, Joey. As we can see on this specific slide, and what Joey has also mentioned a few times when he went through these different recordings, and the steps that we can do is really the tailored function within the tool, is that you can make clear, intuitive figures for each of these different within each of these different tools. By adjusting colors, size, labels, legends, to really make it your own and put your own stamp on that. But also specifically for our commercial clients that have a specific brand, you stick on brand. You have higher impact of your data. So all of this was taken into consideration when this platform was developed, to allow our customers that freedom of making it and giving their own feeling on that. Moving to the next one, exporting data out of these tools is very important for our clients as well. And so it was a high priority to be able to give the option of exporting images, and to export straight into your slide deck or publication, or report that you need to provide. High resolution, easy to read, easy to export images are very important, are a big consideration in this platform. And so with that, our platform really provides high resolution and easy access to these images. They are exported in an SVG format that allows you to retain that high resolution. And then, of course, once again, taking advantage of the customization is very important. So next is really the whole aspect of collaborating and providing an opportunity for clients to engage with their own data, but then also across multiple groups, that the collaborative space that many of our researchers are in. So collaborating with a global community of life sciences is a challenge sometimes, and can be difficult to work with lots of data and big data files. So collaboration becomes even more difficult when you rely on a software that is hosted locally, or an on-site bioinformatician needs to assist with visualization or interpretation. So Metabolon's integrated platform really gives you the ability to collaborate from anywhere. With your data hosted on a secure server in Metabolon's cloud, you can invite users to access your projects and collaborate together. This is especially helpful for multi-centered projects or institutes, or researchers who are traveling a lot and want to make their projects accessible to others while traveling, or just for themselves while on the road, to be able to continually engage with that. So really, with that, it provides you so many additional things. So next is really to where we get to some of the unique tools within our platform. And so we know that using untargeted data comes with a lot of information. So knowing where to start sometimes within this metabolomics data can be quite difficult. Metabolon's new platform has what we now have called biomarker lenses, and they are designed to give you this starting point, curated using publicly available data and thoroughly validated by our expert discovery and translational scientist team. Our biomarker lenses enable you to filter your own data to narrow in on known analytes of significance to a specific disease or biological function. Once your data is filtered, navigate to all the additional visualization tools within Metabolon's integrated platform to gain contextual insights to your data from the perspective of your chosen biomarker lens. And Joey will give a little bit in a few slides. We'll give you some insights into how to do that more specifically. But this is really where our biomarker lenses become very, very focused, allowing you to delve deeper into specific pathways, diseases, or even custom lenses. And the final tool that I will be talking about today and want to introduce to you is really our pathway mapping tool. This is our second unique tool and the last of our 6 tools in our bioinformatic platform. Pathway analysis is pivotal in bioinformatics as well as systems biology, enabling the examination and interpretation of biological pathways to understand the complex molecular interactions that are within in our biology. Features like enrichment results, explore pathways, and explore associations allow research to dive deeper into dynamic views of pathways. So our tools really provide this to uncover significant pathways, delve into detailed pathways visualization, integrated pathways together with genes and enzymes. And at the end of the day, allow you to explore the relationships between pathways and diseases. These analyses are crucial for discovering biological insights, understanding disease mechanism, and advancing personalized medicine by identifying therapeutic targets, potential biomarkers of disease progression, thereby facilitating hypothesis generation and guiding the development of new therapeutic strategies. And so it's really exciting to pass along to Joey again to provide us some of these insights on how these biomarker lenses and our pathway tools work. And so over to you, Joey.
Thanks, Heino. Yeah, to kind of follow up on something that Heino said that kind of struck me is it's important for us to facilitate collaboration, and we also, I think, kind of want to say that we are collaborating with you, and I think these last two slides are the best exemplification of that, where we're trying to collaborate with you and give you data in an actionable way, and in a way that we've combed through a vast trove of literature, and you can kind of see your metabolites in context. So the Biomarker Lens Explorer really has three parts to it that Heino hinted at. There's the Lens Explorer, so you can see kind of in situ how each of the lenses are. And then you have a custom lens editor, which we think is particularly powerful if there's a specific biomarker that you want to contrast against your existing data set that you've run with us, or if you want to look at it against the public consensus with the other data sets. But it's fairly abstract. So to actually show you the thing in action, I'll walk you through it now. So you can see here that we have a generalized description of what a biomarker lens is, and you can read this and get an idea of what a pathway is versus a disease. And you can see that there are three subtabs there with the pathway, diseases, and custom lenses, as well as this tree representation of the pathways, getting more granular as you progress down the tree. So it's not just amino acids and then subpathways. You can actually interact with this sunburst plot and kind of see what is nitrogen metabolism, and it's composed of smaller elements. We also try to be as transparent as possible. So we're showing you directly what the entities in each of the lenses are. And that's available for either pathways or diseases. And you can see that we have a good amount of disease coverage here, especially as it's linked to metabolites, including what might be most useful here, which is this inflammatory bowel disease lens. You can see also that we have other digestive system disorders that might act similarly, but looking specifically at how our study conforms to the public consensus of inflammatory bowel disease seems like a good place to start. We also have this custom biomarker lens, and I've created two that are a little bit more statistically focused, but it's up to you. You can include whatever metabolites for whatever criteria you like. But for example, very significant in high VIP, or there's a paper that I found that really seems to exemplify inflammatory bowel disease. And I want to see how our data look in contrast to what they've found. Again, you have access to the statistics that our statisticians have provided for you. So you can make the determinations on those metabolites based on several different factors. This is something that will really be available in all parts of the app, especially the Volcano and the clustering part. So, if we look specifically at the inflammatory bowel disease, we see, not super surprising, but that our data line up very, very much with the consensus about what metabolites participate in inflammatory bowel disease. If we want to look specifically at this great paper that we found, we can see that there are some significant contrasts with our disease cohort compared to what they have. And part of that might be that theirs is based on VIP statistics and ours is looking specifically at differential. But it might also be worthwhile to filter our clustering plot based on this and to see how those specific metabolites perform against the clustering that happens within, or look more generally at the disease that the consensus seems to suggest is representative of our disease. Again, you have the same interactions. You're just giving a lot more of a structured framework for viewing some of these really, really difficult to read plots. This extends not just to the disease framework, but if you're really only curious about amino acids or a specific pathway that has a protein or a gene of interest, you can really center on with any level of granularity that you want and cut your data a lot of different ways to move forward and to make some determinations on next steps. So to that end, the place that we typically end with our experiments, and something that I think should be familiar to our customers, and we still feel very strongly in, as Heino said, in providing some of the mechanistic forms of our data and the way that it is biologically contextualized, is to put these things into pathways. And we've put a good amount of effort into building out our pathways so that we're better representing the massive diversity of different biological interactions that can happen in a species. So we've made, first of all, the transition from a direct one-to-one relationship of super pathway to sub-pathway. We're kind of looking at it as a tree now, where you have generalized groups, these big pathways, and then you get smaller and smaller because there are a lot of these intricate effects happening that might span canonical pathways or might just be the edge of a canonical pathway. This is coupled with enrichment results that are shown here in this sunburst plot, where you can see the p-value corresponds to a color and should give you a little bit of interactivity to navigate what's a complex and large set of pathways. And then finally, I'll show you something that's a little bit more abstract, but I think exemplifies some interfacial capacity that only we can offer, which is this associations plot. So moving directly into it, as you can see in the interaction with the app, we're starting with enrichment. So basically using overrepresentation analysis and getting more and more finite as you progress down the plot. You can change the p-value in the fold change to change the enrichment calculation using the ORA, as well as navigate directly to some pathways that seem represented by the metabolites that you might be particularly interested in. For example, I find the arginine metabolism in urea cycle to be both a high p-value, high log p-value, and to be something that I'm interested in as an arginine metabolism nerd. So as I progress through the pathway, you can see that it's sub-selected the pathways that seem that are inherited there. Like I've shown, I think a couple of times, you can send this directly to the Volcano plot to look specifically at this pathway in the Volcano plot and the differential representation. And you can see that though some of those metabolites, at least part of that pathway, seems to be activated in contrast between IBD patients and the control patients. We offer two different sets of pathway visualizations. We have our internally curated pathways based on our historical idea of pathways that you can see here. And then we've also introduced a more textbook-looking pathway that shows some of the features and some of the more intricate features of the plot. So you can see, interactions with portal blood, and as you mouse over each of these things, you can kind of see specific IDs that are associated with them. Or if I mouse over one of the metabolites, you can see the specific identity of the metabolites. We can show or hide options to make it as simplified as possible, because we know that these graphs can be quite overwhelming, and you might really only be interested in the flow of metabolite information along the graph. Tried to introduce a couple of navigable elements, because I know the utility of just going from pathway to pathway and seeing kind of, creating a narrative from different pathways can be quite powerful for making sense of this multivariate data. And then you can arrive at something a little bit more familiar, like the TCA cycle or the Cori cycle, and investigate it that way and change the plots around. Something I don't show here that's worth mentioning is these pathways are fully interactive, so you can drag and drop anything that you want and then download the results and then put those right into your presentations. This last entity is an associations plot showing the information flow between diseases and pathways. I see that kidney disease seems to be overrepresented in our IBD cohort, so maybe I want to add that lens and see what the similarity is between our IBD patient and kidney disease. And I can, restart the narrative with kidney disease instead of IBD, start looking at different indications, and hopefully start to generate a holistic scenario based on that, or exclude metabolites if I know that I don't want to look at metabolites that are important to the kidney. Again, we can go right back through hierarchical clustering, and now we're looking at kidney disease in a clustering methodology. So I hope that gives you a little bit of a taste for, you know, the ongoing interactions that are possible even with one data set. And I timed myself earlier, and it took me about 15 minutes to buzz through these pathways. So I think, you know, it's reasonable to assume that within the first 15 minutes of interacting, you can have an idea of some metabolites that are particularly interesting, some pathways that you want to target, and if the perturbations are performing based on literature or based on your expectation, which I think makes this quite powerful and really applicable to a vast majority of data sets. So I will pass it back to Heino to kind of add some closing remarks, and thanks again.
Thank you, Joey. Yeah, so we're at the end of our webinar today. So as we conclude today's webinar, I want to express firstly our sincere gratitude for your participation. We explored today our cutting edge analytical capabilities and demonstrated the extensive support we provide to our clients, enabling them to generate meaningful and actionable metabolomics data. It was also a pleasure to unveil the next phase of enhancing our clients' experience with our data, offering not only interpretive reports like we have done in the past, but also a suite of bioinformatics tools now that allow our clients to unlock the full potential of their data. We hope that today's session has inspired you, ignited some excitement about the future of metabolomics, and also elevated your expectation of your next study. We enjoyed this very much and are looking forward to connecting again, either virtually or at the conference near you, to demonstrate firstly our world-leading data and what other researchers have done with that, but also our newly launched integrated bioinformatics platform. If you ask any questions in the Q&A, we will respond to you as quickly as possible. And if you need to ask any additional questions, please submit an inquiry, and a member of our team will be in touch for a one-on-one follow-up. And then we can take you through both our data capabilities in more detail and our bioinformatics tool, and give you a live demo of that as we can then discuss needs and specific outcomes from your study. And with that, we can go to the last slide, and mainly to say thank you for listening. And with that, I really want to thank everyone for your attention and for the opportunity to share this exciting new development at Metabolon. This is, of course, exciting to share this with our current clients that we have, and of course for all those other clients that will come in the future. Thank you for your time, and we'll see you soon.